Thank you, Dr. Bauman.
Thank you so much, Dean. Thank you so much for the invite this evening. Thank you, Sarah, for that wonderful land acknowledgement. And I feel very lucky and fortunate to be able to work in such a complex and fascinating ecosystem, and also so pleased to be able to interact with the Lower Elwha tribe any chance I get. So thank you again, Sarah, for that lovely land acknowledgement prior to this talk. So yes, thank you all for joining me today. I would like to just introduce myself again. My name is Jenise Bauman. I'm with Western Washington University. I'm with the College of the Environment, and we kind of have a new name here. It's the Salish Sea region, because I'd like to point out that we are well beyond Bellingham and we're encroaching out on the Salish Sea. I know we have lots and lots of Salish Sea to go, but this is a great starter. We have some programming in the San Juans, Everett, here in the Kitsap Peninsula. I'm in Paulsbo now, and of course our site out at Port Angeles, coupled with Peninsula College. So again, our programming goes beyond Bellingham, and I'm just so pleased to be able to talk with you all tonight about the work that me and my students have been doing out in the Port Angeles area. I'm also academic program director for this program, associate professor for the environment sciences, and as Dean said, my research interests are within the plant and microbial interactions within ecological restoration. So I'll talk to you a little more about that tonight. So before I do, I'd love to acknowledge my collaborators, which include two graduate students here, James Kardouni and Andy LeBay, and also my research collaborator, Dr. Rebecca Bunn. And we have been looking at this system here with regard to forest restoration along the Elwha River. So I feel I'm such a small fry in a huge river system. So there are a lot of neat researchers and projects out there. And again, back to my initial comment of how fortunate I feel to be a part of this study system. So tonight, the talk I'll be giving will be on forest restoration on the exposed sediments along the Elwha River, assessing riverbank lupin's influence on conifer growth and mycorrhizal fungi. So if you haven't heard of mycorrhizal fungi yet, you will tonight, and I'll make sure to define all of these terms as this talk unfolds. So again, my interest would be on the forest aspect of the Elwha River. And as you folks are probably well aware, that really to have a positive and successful river restoration venture, you have to really make sure you go hand in hand with that adjacent vegetation in order to get that full recovery throughout this river corridor system. So again, my interests are in these developing forests that you see here. So this is Lake Mills. If you haven't been up here lately, please make the hike. It's a great literal walk through the park right up the Olympic Hot Springs Road to this beautiful vista that expands it and really shows you the enormity of what this lake must have been when it was dammed. And of course, now you're seeing a very dynamic river channel. In fact, I've never seen the same channel twice. Every time I'm up there, something has changed about the system. So my research then is interested in looking how these forests develop after dam removal, and we hope that some of our observations and research can help future projects that are looking at similar ventures out there on the restoration realm. So when we kind of focus up close on the restoration site, actually, let me bump back here. I'm looking actually literally at this terrace in this next visual here, just to show the two contrasting substrates out there on the Elwha, and how these two contrasting substrates definitely drive some contrasting results. So in areas that have documented fine substrate, this would be sediment that has a lot of organic matter, some fine soil, better water holding capacity and whatnot. We did see a really interesting and fast revegetation happen very naturally through natural recruitment within this area. The major players within that growing forest stand would be your Sitka willow, cottonwood, and also your red alder. And in the understory, a lot of horsetail came in very, very quickly initially within those first few years of restoration. This photo was taken in 2017, and you can really see the contrast in areas that have this coarse soil substrate. Very, very rocky, a lot of gravel, a lot of cobble, and a lot of sand. Very little organic matter, very little microbial activity, and just a tough area for plants to try to get established and really grow within these soils. So again, thinking back on the sort of so what big picture, well, if you want to restore healthy water systems, it's really important that you do have the vegetation in place that can really anchor in the remaining substrate, or I would say the sediment that was behind that dam. So there's just not this constant immense erosion. Erosion is going to be a normal part of the system, but you want to try to hold some of that substrate in that river valley. And you also definitely want to get a nice, diverse, herbaceous community that will eventually succeed into forests that will stabilize the land, create some shade, filter the water, and really breed these ecosystem services that will eventually grow into forest stands and provide this nice large wood that will eventually tumble into that river system. So the forests themselves are quite an important component of that river restoration project. So again, some of these major players that recruited in very, very quickly were this red alder. And I like to note that the red alder is so interesting because it's a nitrogen fixer, meaning that it has a nitrogen fixing bacteria that's associated with its root systems. And if you've ever dug around alder, you may have kicked up some of these nodules. They're bright orange, and inside of them, there are many bacterial colonies that are taking atmospheric nitrogen out of the air, rearranging those molecules to a bioavailable form of nitrogen that the plant can readily uptake. So it's an amazing pioneer species that comes in these really nitrogen poor soils and aids in that soil development. Along with red alder came the black cottonwood. They're both very, very fast growers and really prolific with regard to their seed set, and particularly their establishment within wet soils. And upon lake drawdown, where all of that fine sediment was really holding a lot of water in the first year or two, these alders and cottonwoods and willows came in very, very quickly and were able to colonize. And also, again, your Sitka willow, they're in that sort of mix of those three trios that are known to be pioneering species, woody species within the Pacific Northwest forest in Western Washington. So again, just kind of looking at that substrate difference, that literal line in the sand of successful establishment through natural recruitment and difficult problem soils that the Olympic National Park, Josh Chenoweth, really directed that large scale re-veg effort. So you can see a lot of plants that were planted along this coarse soils here. But these coarse soils, or what my graduate student James Kardouni called is these novel terraces that are far removed from the floodplain. So they're not really even technically riparian at this point because they really have no relationship with that river or that hyporheic zone or that groundwater. They're just perched far above the water table, and they are definitely difficult to revegetate because there's just nothing there that a plant needs. No water holding, no nutrients, no microbial interactions happening in these coarse soils, creating a very difficult landscape to actually grow plants. And some folks have asked me, well, why even restore? I mean, eventually won't nature take care of itself? And I guess eventually, and we have to think of what time scale we're working on. If we're thinking of restoring a forest in 100 years, 200 years, 300 years, okay, that's one time scale that maybe eventually nature will take care of herself and re-veg. But if we're thinking that our goal is the restoration of this river channel to be a salmon-bearing habitat once again, is waiting, a 20-year delay, a 50-year delay? Is that okay when your goal also in restoration is really that river channel? So it was important that the park had this big initiative for revegetation, and that there were some novel techniques used to really accelerate forest succession within these landscapes. So here's James Kardouni. We're mapping out his plots along the Elwha River, and the overarching question foundation of James's research was, well, how can we accelerate forest succession in these soils that don't really have the profile to really grow a healthy plant? And let me just pause really quick and define what I mean by forest succession. So simply put, forest succession is really just a change in plant community over time. So that could be in this primary successional stage where you're starting out with no soil, essentially bare rock, and you're going to have a very different timeline as well as pioneering community within this rock substrate than you would if you had this sort of secondary succession, which would be succession starting out with soil. So if I just kind of bump back once, the Elwha was such an intricate and unique system because you did have some soil present on the valley walls, and you saw plant succession able to come in very, very quickly. But in situations where we're rock, we're really looking more on this model of primary succession. And how do you speed this process up to actually get soil organic matter put within that profile and really lead the way for this faster change in plant species community composition over time, where you're getting your weedy species and your herbaceous species in faster to allow for the eventual succession of your woody species, which are your shrubs, your small trees, and eventually that climax community of conifers. And of course, forests too, just like that river channel, are very dynamic. So it doesn't necessarily happen in such neat stages. Sometimes it does, but then there's sometimes these secondary disturbances within the system that shakes up the way the plants arrive and really interact with one another. So again, this goal is, how do we speed up this process of succession when in some parts of that river corridor, we're really kind of starting out in this pioneer primary succession where soil is really not there. So how do we speed that up, and what other techniques can we use to help this? So one of the projects that the Olympic National Park and the Lower Elwha tribe started was really led by the tribe, was bringing in via helicopter all of this large woody debris. And the whole goal of this debris was to really create this complexity, this structure within these coarse terraces, in hopes that these structures will create microclimates that are a little more conducive for seedling establishment. So you can see some of these conifers tucked in with intent, planted around this large wood, and hopefully this large wood will kind of guard against really that wind. It's a very windy system up there in that corridor. Hopefully will cast some shade, can help with water relations with the plant. And at first, when James and I were setting up, we're like, it would be really neat to see what influence this wood has on the establishment of plants and their mycorrhizal partners. So that was what we were kind of thinking of when we were going in, and we were looking at the sites, and we took note of this lupin that we're growing. And then we went back up there to really start marking out plots, and we couldn't even find our tree. We couldn't find the large wood because the lupin really exploded. So if you guys are familiar with this system, you may have been up here when this lupin plant is in bloom. It's very, very beautiful. The bees are drunk with craze and pollen, and it's a really neat natural setting to behold. So lupin was seeded in by the Olympic National Park because this plant here was one of the first pioneering species to come in after Mount St. Helens exploded. So this propensity, or this ability for lupin to come into very disturbed soils that really aren't soils, that are really rocky substrates, and establish was really what they were thinking. Well, let's mix in and lupin with some of these seed mixes. But the fact they did so well was really quite interesting. Another thing about lupin is, like the red alder, lupin too is a nitrogen fixer. If you ever pull up the roots, look real closely, you'll see these nodules growing off those root systems, again with bacteria within those nodules that aid in that transformation of atmospheric nitrogen to a bioavailable form. So we quickly were like, okay, forget the large wood. What does lupin, how does that impact those seedlings that were planted by the park, because in a way you're like, gee, it's a good thing, but it's too much of a good thing, too much, or we're going to have sort of this competition issue where lupin is just smothering conifers, or are we going to see a beneficial attribute, even though in some cases this lupin came in very, very dense, and that was just dependent on how much it was involved in that initial seed mix. So in some areas this was quite dense. So then our research questions became, well, let's check it out. What is lupin's influence on the restoration conifer planting? So the conifers that we were using or were looking at out there were Douglas fir, grand fir, and western white pine. So we had those three conifers that we tagged throughout those terraces, and we wanted to understand how this lupin is interacting with these restoration conifers. We also wanted to know, okay, well, we know it's a nitrogen fixer, but what is that impact of nitrogen within that soil substrate and within the conifer foliage itself? So when I talk about the foliage, I'm talking about the needles of the conifer. And then lastly, we asked the question, well, what is the influence of lupin with regard to the mycorrhizal development of these conifers? And I'd like to take a little time to define what I mean by mycorrhizae. So mycorrhizae is a symbiotic relationship between a tree and a fungus, and it is the norm in our forest settings. It's the norm in our meadows too. Most plants are going to be mycorrhizal, or some sort of mycorrhizal. Our trees, definitely, they're going to be strongly mycorrhizal. So essentially, mycorrhizae is fungi that form these specialized roots with trees. And there's benefits there with regard to really nutrient cycling, nutrient absorption, water absorption, protection, all of that. So there's a lot of neat benefits that mycorrhizae does for the plant. And in return, the plant sends the fungi carbon in the form of photosynthate. So it's this nice mutualistic relationship between the tree and the fungus. And as I said, it's the rule in our forest setting, not the exception. Most plants are mycorrhizae. So what we were looking on, because we were looking at our conifers, conifers are going to be strongly ectomycorrhizal. So this is a group of mycorrhizae that's defined by this external or exterior fungal sheath that surrounds the root of that conifer. And you can also note that it has this neat radiating hyphae that really proliferates throughout that soil profile. So the benefits then would be greater access to water, because fungal filaments are much smaller than even your finer root hairs. So it can access these micropores in the soils that plants cannot. It also can allow the plant greater access to nitrogen. So nitrogen is one of the most limiting macronutrient within the Olympics, and it's particularly limiting in these new soils. So therefore, if you have a partner that can really scavenge nitrogen out of organic matter, even though there's just a little bit of organic matter out there to be scavenged, that could be quite the benefit. So that's going to scavenge nitrogen, feed itself first, and give the rest to the plant. It also, mycorrhizae, will provide an already established network. So what you're seeing in this picture here, the gold is the actual plant tissue, but this white is fungi. It's all fungal hyphae. Check this out. Look at this little tiny seedling that's just germinating, and boom, it taps right in to an already existing mycorrhizal network where it can receive water and nitrogen, other nutrients within the soil profile, and interestingly enough, can receive carbon from these larger trees. So this would be considered a little sink, a little growing seedling that will receive carbon from a source, which would be a larger tree that is involved in that mycorrhizal network. So it's very, very beneficial and very important when you think of mycorrhizae and restoration, because it can buffer transplant shock. A lot of times, if you're out planting plants from field nurseries or seed nurseries, they're going to be mycorrhizal because it's the rule, not the exception. So most of your plants, and in fact, in order to get rid of mycorrhizae, you kind of have to use fungicide in order to clean roots from that. So most of your plants, even coming from that field nursery, will be mycorrhizal, and it's important because it could buffer transplant shock upon planting. So it really is quite an important relationship. And like I said before, mutualistic to both partners. So all the goodies that the plant receives in return, it gives it a niche. So it has a home. Fungi can be housed within these root systems. And of course, they're going to receive carbon from that plant in the form of photosynthate. So it's a nice mutualistic relationship, but always question mutualisms. They too can kind of swing on a continuum. And at what part can a mutualism become either neutral or parasitic? That's also out there in the literature. So, but for the most part, when everything is balanced, there's some type of deficiency, some type of limiting resource, your mutualistic partner, the fungus, becomes very, very important in establishment. Okay, so we wanted to test then the influence that lupin has on our conifers. So to test this, we selected 24 study plots all along the western side, or I guess these western terraces along that former Lake Mills lake bed. And we defined 3 treatments. We had within these treatments 8 plots, very dense lupin, kind of a lot like the photo I showed you before, just a lot of lupin. The other treatment plot, they had a medium amount of lupin, kind of a Goldilocks, kind of just right. You still could see the plant. It wasn't quite buried, but they were neighboring lupin very, very closely. And then we also selected treatment plots that had very sparse lupin within those terraces. Again, 3 conifer tree species were selected, and 3 conifer trees per plot were sampled. So we had a total of 72 conifers that we sampled for both growth, nitrogen, and ectomycorrhizal colonization. And this is what we what we saw as sparse. So really there was hardly any lupin in our sparse plot. So we didn't have anything really neighboring, nothing really influencing regarding lupin in our sparse plots. In our medium plots, again, we sample in the wintertime. So we sample our roots when the tree's dormant, and there's very it's non-destructive technique. So when we're actually taking these photos, we're kicking ourselves like, oh, I wish we had this during the growing season. But you can see where the lupin is dying back, because this is a biennial here. So we did have neighboring lupin neighboring all of our conifers within these medium plots. And of course, these dense plots were just very, very thick with lupin density. There's me out there taking some measurements or some points. And again, some of this lupin was waist high and like I said, very, very thick. So you kind of question, like, oh man, are you okay down there? A little conifer kind of growing within the lupin. So we wanted to take a look at just the influence these different densities had on conifer growth. We also sampled everything from mycorrhizae, and to do so, like I said, we did in the winter, well, the conifers were dormant, fast asleep. So we just trenched in very gently to take our samples off the root system. And then once we got the samples, we took them back to the lab, cleaned them up, quantified. So we're counting mycorrhizal tips. You see these white covered root tips. Those are mycorrhizae. So we can get an idea how much mycorrhizal or how much colonization was there within these field sites. And then we also took it a step further. We extracted fungal DNA, and we used these fungal specific primers to do so. So what this does here, it targets the fungal genome at the 5.8 ribosomal RNA gene. And this is what all fungi are going to really share. There's going to be very close similarity between all of our fungal species, can kind of root all within this 5.8 ribosomal RNA gene. Now, what makes this whole segment so robust with regard to a barcode of identifying fungi molecularly is these ITS regions, which stand for internal transcribed spacer units. They don't code for anything, and that's key. They just kind of space the genes apart. So if mutations occur within these internal transcribed spacer units, it's not going to kill the fungus. They're going to live just fine. They're just going to have mutations with this internal transcribed spacer unit. So different species through evolutionary time have built up enough mutations to where if we took this snapshot of this fungal DNA segment, we can root them all. So they're all similar with regard to this fungal gene here, but there's enough mutations on this internal transcribed spacer unit to where we can actually read the sequence and differentiate among our species of fungi that are out in the field. So it's really neat. It's a neat concept, specific primers that allow for that barcode for molecular identification of fungi. So we sampled, we saw, we quantified, and we confirmed using this extraction technique. And this is what we found. So within lupin's influence, again, that's our question, what's the influence on our conifers? I just want to note on our x-axis of all of these graphs here, you'll see our treatments, dense, medium, and sparse. Okay, that just means the amount of lupin there. Our first panel here is tree growth. So what we saw is that the conifers growing in the medium density of lupin, they were really quite big. I mean, they had the better growth, they're larger trees, similar to the trees that we saw in the dense plots, although this seemed to be our Goldilocks effect, right? So we had our bigger trees in our medium plots, and they were significantly larger than those trees growing within the sparse plots. When we took a look at foliar nitrogen, so the amount of nitrogen that was bound within those conifer needles, what we found too is within the dense and the medium lupin plots, we had an increase, a significant increase in foliar nutrient nitrogen within those conifers, and that was similar with regard to our lupin treatments and significantly higher when compared to the trees that were growing in those sparse plots. So that was devoid of any type of lupin. And our last panel here is looking at mycorrhizae. So this really interested us. So when we saw after quantifying our roots, we had significantly more mycorrhizae in the sparse plots when compared to the medium and dense. In fact, we didn't have much at all mycorrhizae-wise in our dense or medium plots. So it seemed like mycorrhizal colonization on the roots, which we know as this beneficial mutualism, was maximized under the sparse lupin conditions and significantly decreased when growing within those lupin canopies. So we determined a list of species through our DNA sequencing. The most common species that we saw out there was Thellophora terrestris and Wilcoxinia amilcoli. These both are really ubiquitous fungal species. You'll find them everywhere. You'll find them in greenhouses. We kind of suspect that maybe they were brought in upon outplanting from the greenhouse, or regardless, tough stuff. They're able to survive within these coarse sediments and really create this functional mycorrhizae on these plant roots here. We also saw some Cilius, tuber, hebeloma, and rhizopogon, but barely. We just had one hit from rhizopogon, and that is an important symbiont to Douglas fir, and that was one of the plants that we looked at. So without that symbiont there, you're kind of concerned about that mutualism not occurring and whether or not those firs are going to struggle. But in our case, if those Douglas firs were growing within lupin, they didn't have mycorrhizae and they were growing larger and they had plenty of nitrogen. So that was pretty interesting too. And then we also sequenced just some, I don't know, we're calling them little pathogens. They didn't seem to be that harmful, and some endophytes. They were just other fungi that live inside the plant root. We're not quite sure of their function at this time, but we wanted to go ahead and report those data. So we found a small list of species, too, that may have come in with the greenhouse plants, and a handful that probably represent some early successional pioneering fungal species. They, too, will go through a below ground succession as that plant community develops above ground. So we're kind of scratching our head with that reduction of mycorrhizae within the lupin canopies. And we're just kind of thinking, well, if we go back to sort of this beneficial mutualism of this greater access to nitrogen, that's such a bonus for mycorrhizae, but because it's mutualistic, it's going to cost you a little photosynthesis cash, right? So it's not happening. This trade-off is not happening for free. So we're kind of thinking if nitrogen here is plenty in that system, when we saw that pattern in the foliage, could the plant be sort of shutting off that relationship between that partner, that fungal partner? Therefore, they're getting their nitrogen for free and not having to pay that partner for that carbon. We're not sure. So that led into a summary with regard to us thinking about, okay, well, what's going on in the field here? We saw this growth in nitrogen increase and this decrease in mycorrhizae and these sediments. We also are tying it into some previous research done by Cortese and Bunn that showed sites like the Elwha tend to lack fungal propagules and organic matter. And that's going to limit the formation of these functional mycorrhizae. So we're kind of thinking that, okay, boy, lupin could be a real great companion because perhaps it's relaxing this need for this mycorrhizal symbiosis. It might even be programming something with the plant to have it sort of relax its need for that mycorrhizal partner, because the nitrogen is so plentiful around these lupin species. So it got us kind of thinking, well, gee, what was that mechanism? Was it nitrogen that really did allow for this decrease in mycorrhizae within the field? So this introduces my next graduate student here, Andy LeBay, and he's in progress now. And he's taken a look at grand fir seedlings in greenhouse treatment. So he has 4 treatments that he set up. So again, we want to test to see if we're seeing this decrease of mycorrhizae again with this companion planting with lupin. So we also added a nitrogen treatment. So let me just go through all of the treatments here in our greenhouse study. We had a control, and that was just simply a grand fir growing all by itself and just receiving the same water regime as all the other seedlings here. We also had a grand fir that was planted with lupin. Check this out. Lupin got big during our study here. We grew lupin from seed and then would transfer the plugs into our conifer plant. We also had the pink flags here indicate a grand fir that received a synthetic nitrogen treatment. So this was pretty regular. It got watered with a little bit of nitrogen just to kind of keep adding nitrogen into the system. We're hoping we were mocking this ability to add nitrogen in, similar to what we think is going on with lupin. So we had that synthetic nitrogen treatment. And then we had this competition with non-nitrogen fixing plants. So we grew a little bit of Oregon sunshine with, I think this is blue wild rye, and that is called our competition treatment. We wanted something there with the root system, some type of competition that was going on that just didn't add nitrogen to the system, so we can compare those behaviors that we're seeing within the plant or within that root system and alongside of all the other treatments. So again, there's 4 treatments within this greenhouse led by Andy LeBay, and I was lucky enough to team up with Rebecca Bunn, and it was really her help and direction within this greenhouse that really made for such a neat study here. So this was planted in the spring of 2021, so we got everything established in March, and we let it grow and fertilize until November. So in November, we pulled all of our seedlings, took all of our measurements, and these are those results. So looking at growth, what we did not find was any difference between our treatments regarding growth. And this would be regardless of height, regardless of basal diameter. We took biomass, root shoot ratios, the whole thing, and we couldn't find any differences upon any of our treatments regarding actual conifer growth. So this is just showing height, but this was a similar pattern regardless of that growth measurement that we actually took. Again, our x-axis is here. We have our control plots. Our lupin plots, those are grand fir planted with a lupin, grand fir receiving synthetic nitrogen, and our grand fir that was under competition with non-nitrogen fixing plants. So in this case, no difference in plant growth was noted. When we looked at mycorrhizae, again, same treatment on my x-axis here, control, just grand fir growing by itself, lupin, nitrogen, and competition. We did see a significant decrease in mycorrhizal roots in the nitrogen treatment. However, we did not see any differences when we looked at control and lupin or compared that to the competition. They were all really similar. And do note, this was much more mycorrhizae than we saw actually in the field site. So we had just much more mycorrhizae there. Grand firs, they're a little more obligate than other species. So they're really great indicators of that mycorrhizal symbiosis, and will show a nice sensitivity if we do have a decrease. And the only decrease we saw in our mycorrhizae on those roots was in response to our synthetic nitrogen treatment. So we did see nitrogen as a driver that reduced mycorrhizae on the roots. We went ahead and confirmed what we were seeing, again, using this DNA extraction technique. Didn't find many species, so only found really 4 different types of species there. Our primary species were the same primary species that we found in the field site, was this Wilcoxinia and Thelaphora species, and they were our greatest. So that was about 80% of our trees were colonized by these 2 species of fungi. We also try to compare all of our treatments regarding to the dominant species, and we really didn't see a difference there. And that's probably because this Wilcoxinia and Thelaphora terrestris was just the majority, the main species within that greenhouse community. So again, we're still kind of thinking that these 2 probably came from the greenhouse. We did not sample the greenhouse plants. We have since then, and they're in the freezer ready for some mycorrhizae action to kind of confirm possibly these are just greenhouse species that are picked up there. And wow, again, hats off to these species. So not only are they colonized under nicer conditions within the greenhouse or the field nursery, but they're also maintaining that mycorrhizae in sediment, because I do want to note that these plants here, they were into different treatments, but they all were planted using the sediment that we collected along that river. So again, very little. Or could these be 2 primary species that are able to kind of blow into those sediments? We're still kind of working out that detail there. So again, big point was few species we found, no differences among our treatments, but we did confirm what we were calling mycorrhizae were indeed mycorrhizal. So when we took a look at foliar nitrogen, again, same control lupin nitrogen in competition on my x-axis, looking at the amount of % nitrogen in conifer needles here, we found something that was a bit interesting, or at least we think so. We had significantly more nitrogen with our lupin treatment and with our competition treatment. And that was statistically higher than the foliar nitrogen associated even with that nitrogen treatment and with our control treatment. So there were some similarities here. So again, significantly higher foliar nitrogen found in the plants growing within that lupin treatment and within that competition treatment. And I just want to remind you, there were no nitrogen fixers within that competition treatment there. Okay, so just to summarize what we found here in the greenhouse and start making connections of what we're seeing in the field, lupin in this case did not have a beneficial influence on conifer growth. And we're not sure if we're kind of thinking, well, under greenhouse conditions, maybe there was a little more stress with competition than the benefit of nitrogen, and everything just sort of eventually by the end of that study, that greenhouse study, we had a lot of packed root systems within our competition and our lupin treatments here. We did show that synthetic nitrogen will decrease the amount of ECM on conifer roots. So that mechanism, which was theorized, also seen in other greenhouse studies, also played out true in our study as well. We also saw this kind of interesting result of lupin and the competition treatment had increased foliar nitrogen. And we're still chewing on this a little bit, but the only thing that we can kind of use to explain this is that all of our seedlings, we wonder, I don't know if they had some slow release fertilizer associated with that sort of root ball upon planting, so that as they're growing within these different treatments, we just have an increased leaching effect of nitrogen through that sediment, when there isn't a competing root system there to really slow that ability to leach, for that nitrogen to leach out. We think we're just chewing on that result, but it leaves us to question, well, why no decrease then in ECM and lupin like we saw in the field? Why did that not happen in the greenhouse? And why did we not see really any decrease of that mycorrhizae with the competition treatments, even though we see this evidence of increased nitrogen? So we're kind of thinking this. Companion plants are context dependent. In a system where if nitrogen is your limiting resource, but you're getting plenty of it, well, maybe it's time to shut off that relationship by that mycorrhizal partner and keep the carbon for yourself. However, when you have this situation where you're growing in the greenhouse, you're not in the field, you're in a pot. Therefore, that competition between you and that substrate is going to be on. So that may have provided enough stress where that plant felt the need to maintain that mycorrhizal partner in a situation of extreme competition. So how do field plants balance that below ground competition? Well, the way I see it, it's time to head back to the field and take a look at root architecture within these sediments. So that will be our eventual next step in really understanding this neat interaction between lupin and your conifers, and how all of that plays out in long-term forest succession. So if we take a step back really quick and kind of get back onto our observation deck here and think of the big picture of the study, and most definitely the data that we hope that we can synthesize here, we can synthesize that into best management practices for accelerating revegetation projects where dam removal, in this case and other future cases here, coupled with reforestation, will restore the forest and river processes required to structure that wildlife habitat, which not only includes these great forested corridors throughout these river systems, but also how that translates into healthy waters within that river system. So again, we're hoping to continue monitoring out there and to really understand that interaction between companion plantings on these coarse soils that we see. So please let me know if you have questions. Here's my email address. Feel free to shoot me an email here. I would definitely like to thank my graduate students, James Kardouni and Andy LeBay. They've worked very, very hard with me to come up with these data points that I discussed today. Also, I want to thank my research collaborator, Dr. Rebecca Bunn. She's also had some work here at the Elwha, and she really helped us set up that greenhouse study in Bellingham. So I'm here down on the peninsulas, so it was great to have her support and her eyes on that greenhouse project as well. Research support, couldn't have done it without Josh Chenoweth. He was one of my first contacts when relocating here, and was very, very wonderful with regard to access and explanations, and really helped mentoring me understand this system. So I'm very grateful. Also, Dr. Robin Matthews, Dr. Jim Hellfield, they helped on a research angle. Brian Kasak, he's a student at Western Washington's Peninsula College campus, who also helped on some of the DNA extractions. Thank you, the Stunts Foundation and Western Washington, for funding. So hopefully, I think I have plenty of time to take some questions, and I'm happy to. So thank you guys for your time and attention tonight.
Thank you very much, Dr. Bauman. That was fascinating. We do have a couple of questions to get us started, and I'm sure more will be coming in. So if you do have questions, go ahead and type them into the chat, or if you'd prefer, you can just say, I have a question, and we can call on you, and you can unmute and ask them as well. So one of the first questions came in from one of your early slides, which showed some of the pioneering species, the red alder and the cottonwood and the Sitka willow. And there was an asterisk there, and it said that there was a recent dieback of the red alder. And so the question from Janet is, why is there the alder dieback?
That's a great question. I think what we're seeing is, again, this is not quite riparian, just because we're so far removed from the actual floodplain and that underground hyporheic zone, that sort of underground water system that will influence your floodplain. So that's the nuance. We're up here. So upon drawdown, this really did play out like you would see succession on a floodplain. A disturbance occurred, you have wet soils, your pioneering floodplain species came in very, very quickly. Red alders, part of that trio. Well, there's more, but for the main part, they'll come in very, very quickly at drawdown. Now, what you don't have is that reconnection or that constant connection to the floodplain. So therefore, you're going to have your soils begin to dry out as time moves on. So you'll note that dieback of that alder in some of these scenarios. Now, you still have the cottonwoods. Cottonwoods, in contrast, they'll come in very, very quickly in a system, but their first few years, they're just focused on deep rooting, and they'll root and root and root and root until they really tap into underground water. So once those soils dry, those cottonwoods will maintain that connection. So I think what we're seeing is just some natural drying patterns happening post water or reservoir drawdown. That's just kind of selecting those alders out of the system. And then what you're seeing understory are some conifers, some that were planted by the park, some conifers that are moving in very, very naturally. So again, that's part of that successional trajectory. On a floodplain, those alders will be there for decades, right? Although they're not long-lived species. I like to think of alders as those fast-growing, live fast, die hard kind of species. Well, they'll come in, they'll put on a lot of biomass. Eventually, they'll succumb to either a disturbance or a short life, or they'll blow down and reveal an understory of conifers that have established underneath that canopy, and they'll release those conifers. So again, thinking of successional trajectories, some things I don't want to say are predictable, but what we do know about our floodplains is that after a river disturbance, you'll normally have those tree species move in really quite quickly and be able to maintain their lifespan a little longer because they have that constant connection to some wetter soils than they would up on these terraces.
Yeah, thanks. They also were quite numerous. I mean, they came in quite the numbers. So just probably competition between them for those resources, right, would lead to some of them.
Thank you, Dean. And we can't forget that, right? Competitive exclusion is also playing out. And I think it just happens a little faster when you do have that major resource that's sort of dwindling. In that case, the available water, because they bring their own nitrogen, right? So they don't need that, but they do need that wetter soil. Great point, Dean.
Thank you. John asks, you were mentioning the 3 conifer species that you were studying. And John's question relates to the Western Red Cedar. He noticed that they were not doing as well as the other conifers on the lake beds. And his question is, do cedars do better in a more mature environment? So I guess where do they come in that succession process?
Yeah, great question. They'd be considered a little more later successional species. So you're right, they'd be coming up kind of after that alder has been there for decades, and the soil's a little more developed, and there's a little more shade on that system. That's when your cedars would come in kind of on more of a natural successional pattern. But this whole thing was one big ecological hypothesis to test after another. So including them in was also a pretty interesting data point, even though we're not surprised they didn't do so hot, but some have actually established, and now I'm seeing they're under some pretty harsh brows. But yeah, you're right. I think on a successionary trajectory, they're a little later than those pioneer species.
Awesome. So Paige asked, are other plants aside from lupin being considered for the next stages of the study?
That's a great question. So I guess just to clarify, do you mean the study that we're looking at, or the work that they're doing in the park? And I can kind of talk a little more about our work more so than what's going on the park aspect, planting wise.
I don't see. Yes, your work, Paige.
Oh, okay. Thank you for that. In fact, we look, we're doing a similar study. Actually, we're writing it up now on what we're seeing in the Appalachian coal mine. So it's kind of a neat juxtaposition, these two projects that I have. One are just desolation of Appalachia forests and this sort of reconstruction after coal mining. And this was this total, moonscape from the drawdown and that reconstruction, and they're going in very different directions, as you could probably guess, because you have more of just a natural puzzle pieces here in the system than you would do in Appalachia. So what we're seeing there are some invasive species that come in very, very quickly on mine sites, and that's because they were erroneously, and again, hindsight's 2020, used as restoration plants decades ago. So 40 years ago, they seeded all these exotic species. They were great. They worked wonderfully. 40 years down the road, oh my gosh, they're invasive and they're eating all the seedlings. So now we're taking a look at, okay, why? What causes sort of that interaction? And we're looking at a plant called Lesbedeza, and I like to call that Scotch broom's little cousin. It's a legume too, so it's producing nitrogen. And we're finding if you remove compaction out of the coal mine system, Lesbedeza actually doesn't inhibit growth from some seedlings, not all seedlings. So they seem to have a positive reaction, a relationship with some of these trees. And I think it's because it's adding nitrogen, and it's also buffering a little bit of shock, creating a little bit of structure, but that's not the case with all plants. So where we're seeing it, that doesn't seem to interrupt mycorrhizal associations. We've seen tree of heaven. That's another invasive species on the mine sites. And I say that kind of with a little smile, because every time on Peninsula College's campus, I walk by that tree of heaven that's kind of limping along. Believe it or not, they're major invasive species out in East Coast, and they might be working their way to the west as well. So tree of heaven can be very detrimental, and we found that they have a negative impact of mycorrhizal colonization on oak species when you're trying to restore with oak. And then we've also seen some detrimental attributes that Herb Robert has with regard to that mycorrhizal relationship between plants and trees. So thank you, Paige, for that question. Yes, we are looking at different species. So far, they've been all exotics. So to me, it was so interesting to see what a native species and that interaction could possibly play out on a landscape. Because that's what my first thought when I was walking on these terraces. I was like, uh-oh, boy, these lupins are really taking, I wonder if they're going to smother the trees. Josh Chenoweth, he was cool, calm, and collected. He's like, they're going to be okay. I was like, I don't know, Josh, I don't know, but yep, they seem to do rather well. So yeah.
Great. We have some other questions about invasives we'll get to. But the next question is from Emily, and she wants to know, do you have criteria to determine quote success unquote for the area's restoration as you continue to monitor it?
I think I caught that. Did you say what determines success?
Yeah, what is success, I think, is as you continue to monitor.
Gotcha. I think just sort of generically, success would be defined as a system that is resilient and self-sufficient. So if we get to a point where we can see that there really isn't this invasion that's marching in, invasive species aren't marching into this system, you can have disturbance, which is a natural component of your ecosystem, with a healthy rejuvenation of native plants. I believe that would be a successful re-veg for this area. Again, down at the floodplains, you're going to have, again, your disturbances. Floods are going to happen. That's going to be your natural disturbance component on the floodplains. That's okay. So I think on a sediment aspect, maybe those changes are going to continue to occur. And I'm not a fish biologist, and I don't want to pretend to understand that ecology, but I would imagine maybe what would look almost like we have some resiliency on the forest end may not quite be as resilient on a floodplain end. If you're having bursts of sediment that might impair breeding habitat. So I want to just make sure I'm really clear when I say success on a restoration end here, I would be looking at the vegetation community and their ability to rebound post natural disturbance events into that system. And when I say rebound into a native community, that could be maybe again your willows and alders, and this succession just kind of clock starts back, or it could be some blowdown of your alder and a release of your conifers. But those changes that are occurring are gonna resemble some of the reference sites that flank that area.
Great, thank you. Our next question comes from Laura, and she asks, she makes this point. She seems like water scarcity would impact seedling success in the field. And so she's wondering, why wasn't water one of the greenhouse test variables?
Oh, Lord, let me tell you. The water in my tears. So what we, one thing we did do, we had a companion study that we set out on the lower Aldwell area floodplain. We did the same thing except for synthetic nitrogen. We did not use that treatment. We just kept it with lupin, control, and competition. And guess what? I had one survivor after that heat dome effect. So you are right. Water scarcity is another stressor that's really going to create a nuance to the system. And we've talked about this because of two things we've talked about. One, water scarcity, just like you said, that's going to be a stressor. That's going to maintain your mycorrhizal partners as well. And then we're also wondering, okay, what do companion plants do regarding water relations? We know it's going to be a competitor, but kind of this is where I'm ending my slideshow here, is that, okay, it's not going to be a pot competition and they're really competing for the resources, including water. It's probably going to be a root architecture thing where your trees are rooting deeper, your herbaceous plants are a little more fibrous on that surface, and there's this interplay. There's enough space to differentiate niches within that soil profile. Well, where does water come in? You're right, that's going to be a stressor and a scarcity. But the question, so this is something now for a future study, would be, well, when you have these novel systems here and you're trying to restore them, at what point do these companion plants actually aid water relationships and also become competitors for that limiting resource? And what I mean by that is plants will do a funny thing. It's called hydraulic lift. And you'll notice it. So if you're ever in an area like the Elwha or desert areas or arid areas, you're going to see a big tree, and around that big tree, you're going to see little plants growing. It's going to be this sort of island, this facilitation island. Well, what plants will do is they're taking up water. Oh, when they shut their stomatas and the sun's setting, they relax. They release a little bit of water out of their roots, and that hydraulic lift and subsequent release creates this facilitation relationship between a neighboring plant. So my question is, at what point does that shut off and it becomes competition? And that's where you're going to get that water scarcity. So did we see something early enough in restoration where we still have some water in macropores, where there was actually more of a facilitation with these water relationships with the lupin and the fir? And then later on, like now, when I'm trying to establish plants in certain areas, that you get such a water scarcity that mycorrhizae doesn't even help you in the end. So you're right. That water relationship is going to change in various times of relationships with plants, with the relationship with your soil profile, the timing of reservoir drawdown, the timing of floods and whatnot. So that's an excellent point. Water relations and mycorrhizae would be another treatment to move forward with. And we want to, and we also want to look at other species too. But water relations will be another treatment. So thank you for that.
Well, Dr. Bauman, we have one last question. We're a little bit over 8 o'clock. Are you okay?
I am okay. Thank you.
Great. Thank you.
Yeah.
So this last question has to do with herb Robert. There's a lot of it in the Elwha Valley, down lower in the valley. And so Tom is wondering, did you see herb Robert out on the reservoirs when you were out there doing your work?
No, I didn't see them up here
Mhm.
in Mills. And I was actually, we were out there looking for sites a couple weekends ago, and I didn't quite see that influence of or Robert, I know we're still kind of off season. But I do see it kind of in the lower site around the Aldwell. That pressure seems to be there, and also in some shadier areas too.
Mhm.
So it's there. I'm not saying I've never seen. I have seen them, but I think the park has done a tremendous job of staying a little bit ahead on this upper area. Like I said, they're there, and you can see them. You see Scotch broom, but like I said, they right now I haven't seen an overwhelming sort of intrusion up on this upper lake bed. But boy, you take your eyes off it for a second, you'll get that invasion. And Herb Robert is so interesting. There's suggestions in the literature that has antimicrobial properties that will actually kind of sheathe or sever the relationship between a forest tree and their mycorrhizal symbiont. That the data there is hard to really prove, but that's something we're moving forward in our lab, growing out these fungal cultures on plates and introducing certain plant leachates to see if that's thwarting any type of mycorrhizal growth. So more to come. We hope to kind of look on that influence within a laboratory setting. But yeah, they're there. And again, kudos to the park and the tribe for any type of management with these native plants. But that's going to be a threat, particularly in Aldwell.
Yeah. Well, thank you so much, Dr. Bauman.
Thank you, Dean. I appreciate it.
Yeah, thank you so much. And thanks everyone for coming out this evening or joining us.